Tonight's guest speaker, Dr. Krista McCoy, is the Director of Research and Conservation at Florida Oceanographic Society. Dr. McCoy holds a bachelor's degree from Old Dominion University, a master's degree from James Madison University, and a PhD from the University of Florida. Dr. McCoy's research program focuses on understanding the natural protective mechanisms that are activated by toxicants and other stressors, and determining how those mechanisms can be used to protect human and environmental health. Dr. McCoy tonight will be giving us an update on the important work that she and her team have been up to over the last year. It is with my greatest pleasure to introduce tonight's speaker, my friend and colleague, Dr. Krista McCoy.
Thanks, Zach. Can everybody hear me? Yeah? Okay. I'm happy to be here, and I'm excited to tell you about all the work that we've done over the last year. Well, I should say 3 little stories. And there's a whole bunch of stuff I'm not telling you, so you'll have to either come visit us at the center or tune in next year. So let me see. So the 3 things that I decided to focus on tonight start with a discussion of analyzing water quality data that our citizen scientists have been collecting for over 25 years. And then we're going to talk about evaluating oyster restoration. And then the last little story that I'm going to tell you is about the work that we're doing on genetic rescue of seagrasses. So first I'm going to talk about how we are assessing water quality trends in the St. Lucie Estuary and Indian River Lagoon using citizen science. To start with, we all know that coastal development has really increased over the last 100 years. And along with that development comes algae blooms, fish kills, and habitat loss. So the main area that we have been focusing on over the last year is the Indian River Lagoon. And our water quality program occurs in Stewart and the surrounding area of the southern Indian River Lagoon and the St. Lucie Estuary. So the St. Lucie Estuary is important and endangered because the C-44 canal directs water discharges from Lake Okeechobee along with local drainage basin runoff into the St. Lucie Estuary and then through the estuary into the Indian River Lagoon. And it was 25 years ago, this was the inspiration for our citizen scientists at Florida Oceanographic Society to begin trying to monitor the water quality. And so a bunch of citizen science volunteers as well as Florida Oceanographic employees developed the FLOWS program, which stands for Florida Oceanographic Water Ecosystem Surveys. So we have a bunch of volunteers that go out every week and collect water quality data for us, and this has been going on for literally half of my life, which is amazing. So they look at salinity, they measure dissolved oxygen, pH, temperature, visibility, and water depth. They look at weather conditions and write those things down. They tell us about what the tides were like when they collected the water quality data, and they write notes about the surface conditions. We have 10 testing zones, and so they start at the northern part of the map, and they up in the winding North Fork of the St. Lucie Estuary, and they go all the way down the intercoastal waterway towards Jupiter. Within these zones, we have up to 100 sites that have been active across those 25 years. And so each one of these little red dots is where our volunteers go out and take water samples and do this chemistry for us. So we use this data to collect or to create weekly water quality reports, which some of you all might receive, and if you don't and you want them, we can get you those. We can have them emailed to you. We assign letter grades to each of these zones based on parameters related to general ecosystem health. And these reports are shared with the community to let everybody know how the water is doing. And so this one is from January 1st through the 7th, and as Zach discussed, the water quality is pretty good. We can also take this data and look at how the grades here in this black line are related to water basin inflows. And one thing you'll see is here in May, and when the water basin inflow was low, the grade and the health of our estuary and lagoon is very high. And you can see that again over here where there's low inflow, the grade is very high. And then you can notice that those grades decline when we have a lot of runoff. Makes sense. So we have 25 years of data. And so I'll just give you a little background information. So I've been at FOS for just about a year and a couple of months. And so this year in review is exciting for me because it's allowing me to tell you what I've basically done in my first year here and what my team has done. And so when I got to FOS, Nate Wynn, who runs this program, told me about all this data. And so this is amazing because each one of these spreadsheets is an entire year of weekly water quality from all of those little red dots, right? And so I said, we should work that data up. And he said, okay. And so what I'm going to present is that project. And I want you to know that was my role. I said, we should do this. And he said, okay, and he did it. So our objective really was to examine our historical water quality patterns and trends. But before we could do that, we had to evaluate the quality of our citizen science data. And so the first question we asked was, how does the citizen science data compare to other monitoring systems? And there's a few in the area. One is the Indian River Lagoon Observatory Network, IRLAN. It's run by FAU and Harbor Branch, and it was established in 2015. Our citizen science volunteers have been doing this since 1998. The Kilroy Network is run by Team Orca, and it was established in 2021. And the South Florida Water Management District goes out and collects water quality samples once a month. And so the first two are autonomous systems that collect water quality consistently throughout the day. And then the South Florida Water Management District sends people out to collect that water once a month. So these are our sites I showed you. The blue dots that showed up there are where the IRLAN stations are. The purple dots are a little bit hard to see, but there's one here, there's one down here, there's a few over here. Those are the Kilroy networks. And then the green dots are where South Florida Water Management District goes out and samples each month. So one way to ask whether or not our citizen science data matches well with the other systems that are also collecting water quality data is to use a Mantel test. And this is a statistical analysis that is used to calculate correlations or similarities. It provides a value called an R value. That's a correlation coefficient, and it allows you to determine how strong that correlation is. And so an R value of 1 is a perfect correlation that's positive. And so this is where we get that idea of 1-to-1, right? This is a 1-to-1 relationship. So it's a positive correlation. A 0 means there's no correlation. The data is not related at all. And a -1 means that the data are negatively correlated. So as one goes up, the other goes down. And so what we were able to do is for each of those areas, remember there's 10 of them, we said, okay, which one of these other water quality systems are out there, and what can we correlate our data to? And in the first area, area 1, it's only South Florida Water Management District data that's available for us to compare our data to. But in the second region, we can compare our data to all of the different systems. And we can do this for all of our sites. The first thing I want to point out is that there are 2 R values that are quite low. And we think that this is because the Kilroy system has not been set up for very many years, so there's just not a lot of data. But in general, most of our correlation coefficients are quite high. And so 10% of our correlations are above 0.9, 20% are above 0.8, and 74%-ish are above 0.7. So these just, you have to take my word for it, but these are really good correlation coefficients. So we're proud of this data that, you know, the citizen scientists are out there collecting data on a weekly basis, and that data matches with these machines very well. So we did this correlation, we're happy with the results, and then we also wanted to ask, what about the differences in sampling intervals? Our people are going out once a week. The Erlon and those autonomous systems, they're collecting data every hour. And then the South Florida Water Management is collecting data once a month. So there's different amounts of time that are going by between these samples. So what I am showing you here is a graph with temperature on the y-axis. In red is the data from our citizen science volunteers, and that's a weekly measurement. In blue is the Erlon, which is an hourly measurement, and then in green is this monthly measurement. And what you can see is that our citizen scientists are collecting data that is very similar to the Erlon hourly data, which is pretty amazing. So that's for temperature. We have this similar story for dissolved oxygen. Our values are very similar to Erlon. Salinity is also similar, and so we see that our weekly measurements show the same pattern in the data as Erlon, but our pH data isn't so good. So our pH data is, the red is our data, and it's high, and it doesn't show the same pattern as the other 2. And so we need to think about how to address that and what we might do differently in order to start potentially getting better pH values. Now, I just want to throw out there that I'm assuming that the autonomous systems are correct and our data is faulty, but that might not be the case, right? So we need to dig into this a little bit more. Overall, I think our data is really compelling. I think that the people that have been going out for 25 years have been doing a great job. And so the first answer, how does citizen science data compare to other monitoring systems? I'm going to say quite well. So now that we know that our data, that our citizen scientist is collecting matches with other data that's being collected now, we feel confident assuming that the data that they collected 20 years ago was also good. And that's an assumption that we have to make because nobody else was collecting data in the same way back then. And so now we want to ask, how has the St. Lucie Estuary and IRL changed over time? And these are questions that we are currently analyzing the data for, but I'll show you a bit of work on temperature patterns, because that's kind of what I'm most interested in at the moment. And so this is a graph that takes all the data from all the sites and just asks the question, what is a water temperature doing over this vast amount of time? And so what we see is that temperature has increased in the St. Lucie Estuary and IRL over 20 years, 25 years. What's really interesting is that in 2011 was when the seagrass mortalities, vast seagrass mortalities, started to happen. And we all know that it was associated with algae blooms, so harmful algal blooms. But I don't know of anybody who's really thinking about how temperature might have played a role in that. How did the increase in temperature over those years potentially increase the algae or make dealing with the algae more difficult? Another way to look at this data is to organize it based on time. So on the y-axis we have temperature, and then on the x-axis we have months. And so we can look across the year, across all the different dates that we've done this. So this shows 20 years of data. And one thing I want to draw your attention to is how the bottom tends to be bluer, and so earlier time periods, whereas the red is at the top, and those are more current time periods. So what that shows you is that over time, the water is increasing in temperature. And this is especially problematic for the winter months. This is the winter months are warming more than the summer months are. That's what this data suggests. Another thing that was really cool, and Nate totally surprised me with this when he showed me this data, because I did not realize that some of our citizen scientists are so dedicated that they go out after sunset or before sunrise, sometimes to collect this data. And so what that allowed us to do was to select daytime temperature and nighttime temperature. And what we see is that the temperature in the daytime does increase. We can look around 2010, 2011. We see that these temperatures have increased. They're bouncing around a little bit. But what you really see is this increase in nighttime temperatures around 2011. And again, that was this time period when the seagrass started to die. It could be completely coincidental, but it's pretty interesting, and I think it's worth following up on. So I put this little inlay in here because it's to remind myself that I also want to suggest that it might be those nighttime temperatures that are increasing that is driving this relationship of increased temperature. So it might not just be the daytime temperature. That increase might be driven by those nighttime temperatures increasing and not going down as they should once the sun sets. So the key takeaways from this story is that our citizen science data was comparable to other systems, even machines. And temperature is increasing and has increased over the past 25 years, especially in the winter and especially at night. And I just want to say that citizen science is scientific research that the community does. And so I want to thank all of our water quality volunteers and all of our volunteers. Actually, we wouldn't be able to do what we do without you all. This work was done by Nate. There's his email address. I'm happy to answer questions, but he's the expert. So this brings me to vignette number 2, and this is about evaluating oyster restoration. This story starts off with two fantastic scientists, Anna Braswell and Lorraine Simpson. So Lorraine was the director of research and conservation before me, and before she left Florida Oceanographic Society to go to St. Johns River Water Management District, she and Anna developed a research plan to evaluate oyster restoration that Florida Oceanographic Society had done years earlier. Lorraine left, I came in, the grant was funded, and all of a sudden I had this grant funded that I didn't even have to do any work for, which was amazing. And so Nick Curto and Natalia Jaworski have helped with this project. Emily Sermont is a master's student for Anna, and she really ran the whole project. She did her master's degree basically on this oyster project. What I'm going to talk to you about today is the part of the project that we did at FOS and that Natalia led. But before we go there, I just want to give you a little update or a little background on oysters. So oyster larvae undergo complex development. That means that they change shape dramatically when they're developing. So they float around, the eggs are fertilized in the water, and they float around and they change their shape, they metamorphose, kind of like a butterfly, and then they settle. And when they settle, they're called spat. And spat settle on hard substrates. They can't settle on the mud, they won't live. And so a lot of times they settle on other oysters. And the cool thing is that we can see these spats. So this is an oyster shell. Oops, sorry. This is an oyster shell, and if you look here and here, we can see the baby spat that has settled. And this is fantastic because now we can monitor what we call recruitment, or the amount of those babies that are settling. And you'll see this little hole right in the middle of this oyster, and we drill those holes. Well, we don't, our volunteers drill those holes, so that we can string them on these metal wires, and then we hang them, and then we hang them on these oyster teas. And if you go to downtown Stewart, you might see these. And Nick Curto goes out and he collects these, what we call oyster stringers, every month, and he'll replace them with new ones, and he takes the ones that have been out there for a month, and he brings them back to the lab, and he counts every single baby oyster that has landed on those. We take pictures, and we count the live ones, and you can also see where they settled but died. So we count live and dead, and we have that data for every month of the year, and that's been going on for about 5 years. So one of my favorite questions is, who cares? So who cares about oysters, right? I mean, they're yummy, but beyond that, well, oysters are super important for a couple of reasons. They're keystone species. So if you know about the keystone in an arch, if you pull that keystone out, the arch is very likely to collapse. And so when you remove oysters from a system, because of all the jobs that they do to keep the system stable, that those systems can collapse. And they're foundation species, which means that they literally build habitat for other organisms. We call them ecosystem engineers. So they engineer habitat that allow other organisms to survive and to prosper. They provide shelter for organisms, and they reduce wave action. They provide a surface for things to grow on, they provide food, and they provide habitat for little fish that are food. They clean the water so that they can help other things grow, like seagrass, and they stabilize the substrate. So they do a lot. So that's why we should care about them from a nature perspective. But what about personally? So how do oysters help humans? Well, they're yummy food, like I said, but they also are important for shoreline property owners because they reduce coastal erosion. They're important for both recreational and commercial fishers because when oyster reefs are healthy, the ecosystem around them are healthy, and that provides better, larger catches. It's also important for construction workers and producing jobs for biologists because we build these oyster reefs, we restore the oyster reefs, and we monitor them. So it produces jobs as well. So unfortunately, oysters have basically disappeared. There's not very many left in the St. Lucie estuary or the IRL. And actually, oysters have disappeared from many, many places globally. And so here in the IRL St. Lucie estuary, destructive harvesting started in the early 1900s, where they just removed, large pieces of the oyster reefs. In the 40s, people started to recognize that the margins of these oyster reefs were dying. By the 70s, there was massive overharvesting, and this led to a worsening of the water quality because the filters, oysters filter the water. Those filters were literally removed out of the system. By the 80s, there were declines in oyster spat, so those babies weren't there. You remove the adults, the babies aren't going to be there either. And so that was noticed in the 80s. And what was also noticeable with the decline of oysters was severely reduced water quality. And so the oysters weren't there filtering the water and cycling the nutrients. By the 90s, there was further mortality due to continued habitat loss, poor water quality, algae blooms, and also boating impacts. So since then, we've come to the realization that we better do something about this, right? And Florida Oceanographic Society has been one of the leaders in oyster restoration work in this area. And so back about around 2019, before I was at FOS, so I can't take credit for any of this, they developed the Florida Oceanographic Oyster Restoration Program, or FLOR. FOS staff went out and to different local restaurants and collected oyster shells. Those shells were aged on our property and then with lots of volunteers were loaded into bags, and those bags were put out across the St. Lucie estuary and Indian River Lagoon. These are the sites in yellow where these oyster reefs were created. And in the green X's are where we, and when I say we, I mean Nick, monitors these oysters currently. And they've been monitored since the reefs went in. And so this brings me back to Anna and Lorraine. So they wanted to evaluate the oyster reefs that FOS installed because it's great to go and do this kind of stuff and try to help nature rebuild. But if you don't evaluate what you did, you never know if you should do it again or if you should stop doing it or, maybe how to change things so to make the approach better next time. So they wanted to evaluate these reefs. They were interested in looking at wave attenuation, community diversity, so all the different creatures that are living in and around those oyster reefs, and oyster health. So the part that we focused on, and by we I mean Natalia, is oyster health. So Natalia's interested in disease ecology, and so she was the perfect one of our group to dive in and learn about Perkinsus marinus, or what we call dermo disease. It's a protozoan parasite that's known to attack oysters. And so this one on the left is a nice fat oyster that I was going to say looks delicious, but I'm sorry. I can't do it. It is healthy. And B on the right is one that's emaciated, and it's not very healthy, and it has dermo disease. And if you look at these microscopically, you can see the disease in them. So Natalia developed a procedure for being able to count the number of dermo protozoans in the tissue. So just a little bit about dermo. It's a warm water protozoan pathogen. It thrives in higher salinities, and its growth is reduced when the salinity is low, around 9 or so. Oysters can thrive in lower salinities, and they also live at a bit higher salinities. So they live in places where dermo doesn't do well, and they also live in places where dermo can do well. So oyster mortalities are caused by tissue death and blockage of major blood vessels in the oyster. Who knew that oysters had blood vessels? They have a heart too, and it's got 3 chambers. And I had to look that up because I was like, what? Anyway, so the dead and disintegrating oysters release infective stages of this protozoan, and so that it can spread it when those oysters die. So we set up this experiment to look at natural and artificial reefs. So the natural reefs are yellow and the artificial reefs are pink on this figure. And this was done in Fort Pierce and in the St. Lucie Estuary and southern Indian River Lagoon. And we compared dermo between natural and artificial reefs in these areas. And so what I'm showing you here is the probability of dermo infection. So what that means is, if I am an oyster on an artificial reef in the St. Lucie Estuary, here in purple, I have a lower chance of having a dermo infection than if I am an oyster in the Indian River Lagoon. And if we compare these to natural reefs, you can see that the oysters on natural reefs have a lower probability of disease. Now, this graph is showing you the number of dermo bodies within a millimeter squared section of tissue. And yes, Natalia counted all these. And what you can see in the St. Lucie Estuary in purple is that if you have a dermo infection, it's very low in artificial and natural reefs. But that is not true for the IRL. So in the IRL, the number of dermo bodies per millimeter squared in the tissue is much higher than in the St. Lucie Estuary, and it's especially high in those artificial reefs. So oysters on artificial reefs are more likely to have dermo than natural reefs, and the reefs in the IRL have more dermo than in the St. Lucie Estuary, especially those artificial reefs. And so now we have to adaptively manage, right? So now we know this, now we're informed about taking the next steps for building oyster reefs. Placing artificial reefs in areas that are ideal for oysters and less ideal for dermo is likely the best path forward. And one of the things that we need to study is whether or not artificial reefs that are near natural reefs are leading to higher dermo in those natural reefs. Because if that's the case, we might want to remove those artificial reefs, right? So this is, I think that this work is a nice way to highlight the approach that the research department at FOS takes. So we're not just doing restoration, we're trying to understand restoration and understand how to do it the best way that we can. So now we're at our third little vignette, and this is about genetic rescue of seagrass. The first two stories that I told you about were stories that came to me when I came to FOS, things that were already in the works, things that people were doing without much help from me, not very much intellectual, input, right? I just got there and all this cool stuff was happening. This little vignette is my baby. This is what my background is on trying to understand the mechanisms that organisms, both humans and seagrass, use to protect themselves from stress. This is what keeps me up at night. So the motivating question here is, how do we improve seagrass restoration? Because most restored seagrass meadows die. And when I say most, I mean like 93%. So we go out there and we do all this work and we put out all this seagrass. And let me just tell you, growing seagrass is hard. We have volunteers 6 days a week that come in and clean our seagrass tanks. And without them, I don't know what we would do. And so this is hard work, right? And then you put it out there and it dies, and then you literally cry. And so when I got to FOS, my team was working on trying to understand whether using fencing to control herbivory on that new grass to keep the manatees and the turtles out was helpful. And they were also trying to understand if we plant clams and seagrass together, does it help the seagrass get established more quickly, and does it help it to thrive? And I just want to introduce, in addition to my team, Todd Osborne and Mike McCoy are collaborators on this project, and they're super awesome to work with. Todd is from University of Florida, and Mike works at Harbor Branch, FAU Harbor Branch. So I want to tell you just a bit about our facility. And so we have our seagrass nursery where we grow lots of grass for planting out in the IRL or different restoration projects. And over here on the left side of our huge, this is 750,000 gallon aquarium, which is cool. If you guys haven't been to FOS, you need to go. We have all kinds of cool stuff in there. But my part of this is this little section off to your left. And this is our seagrass meadow. I call it the experimental seagrass meadow. It's also referred to as the wet lab. And it's where we grow our grass for restoration projects. And it's also an experimental meadow, meaning that it's a model for seagrass meadows in the wild. And we can do manipulations in there and ask, does adding this creature help the seagrass grow? What about this? So we can do all these experiments in there. It's exciting and it's super fun. The reason why I'm telling you about this area is because when I got to FOS, my team had already set up an experiment using the grass in this area. And I want to point out that this area is filled with shoal grass, but I've put these little colored cartoons of grass to point out that the seagrass might be all the same species, but they're not all the same genetically. And so there's different strains in there because they've been collected at different times from different areas. And so it's not all one clone of grass. And so before I got to FOS, the team had set up a big experiment to look at whether shoal grass grows better when planted with clams and protected by fences. And so to do that experiment, you need treatments that have fences and no fences, and you need grass that's growing with clams and no clams. And they set this up in 2 different places, one near Fort Pierce and one just outside of our backyard, basically, in Florida Oceanographic Society. However, right after I got to my new job, there was a seagrass mass mortality at both of our sites. And so I know this is going to sound crazy, but I saw this as an opportunity. And it was important to me to convince my team that this was an opportunity, because they were heartbroken. You know, I think it was so much work, and then, you know, they're taking me out into the field for the first time to see this, you know, super site, and then the grass is dead. And, you know, it was horrible. And so I said, okay, this is an opportunity, because we just change our question. Our question now becomes, what strains of shoalgrass survived? Which ones were selected for? Because there were little pieces of shoalgrass still living out there. So now is when you should get your phones out and start scrolling and check out, because I'm going to tell you a little bit about genetics. Don't you actually get your phones out. This is going to be awesome. Okay, so this little character is you. Looks just like you, I know. We have trillions of cells in our body, and most of those cells have nuclei, and inside the nuclei are our chromosomes. And those chromosomes are made of DNA. And sections of the DNA are called genes. These are functional areas of our DNA that basically carry inherited information that make us who we are. They are the instructions through which our body follows in order to develop, and they're also instructions that control different traits that we have. So one example is eye color. So eye color is controlled by multiple genes, and these genes control how much brown pigment, called melanin, is laid down in our iris. If you have lots of that pigment, you have brown eyes, and if you have less, you have blue or green eyes. And then other genes add subtle variations in color to make eyes more hazel or more gray. And so seagrass have genes too. And all genes, doesn't matter if you're a gene that's in an eyeball of a human or in a blade of grass, are made of nucleotides. Nucleotides are basically the fundamental building blocks of DNA, like Legos. And so if you have your cell and you have the nucleus inside there, you have your chromosomes, and if you kind of unwind those chromosomes and look at the DNA, you will see these nucleotides that link together and form long chains. Their sequence spells out the genetic code, just like letters form words. And we abbreviate the names of the nucleotides by their first letter, G, C, A, or T. And there's a single nucleotide polymorphism, or difference, is when one of those letters, one nucleotide, is different in the DNA. And believe it or not, I think this is fascinating. We can figure that out with genetic sequencing today. So we can sequence the DNA and know if a single nucleotide is different. Now, we call these SNPs, single nucleotide polymorphisms, and they occur at specific places in the genome. Specific places on that chromosome we'll know has that single difference. So everyone's genetic blueprint is slightly unique, and these differences change our traits, control our disease risk, and how we respond to drugs or toxins or other stressors. And scientists use SNPs to identify populations of different variants, of genetic variants. And so, for example, here on the top, you'll see the general population has this G, and there's a single nucleotide polymorphism in this example that occurs in less than 1% of the people, and it's a T instead of a G. Okay. So I'm telling you that there's single nucleotide differences in the DNA that we can detect, and 2B rad is the way that we detect it. So this is a method of detecting SNPs in different individuals. I'm not going to go into the gory details, but it uses a special enzyme that can cut the DNA and make short sequences that are really easy to sequence. And sequencing just means reading those letters. So if we focus on that bit, we can, for each one of these individuals, 1 through 4, we can read the DNA and we can see those single nucleotide polymorphisms. And so what I want to draw your attention to here is that 1 and 2 are more closely related because their sequences are similar, the same. They're more closely related than either one is to 3 and 4. And we can do this for a whole bunch of creatures, in this case, seagrass. And this is what we call a phylogeny. It's a figure that shows you how closely related things are. The closer they are in that list, the more closely related they are. The further away they are, the less related they are. And another way of looking at this, or another way that we display these phylogenies, is kind of like if you're looking at cards. And I'm showing you this because I'm going to show you our data in this format. Okay, back to seagrass conservation. You can turn your phones off, take your iPods out, and listen again. Okay, so we have this source population in our meadow that I told you about. And then we have these survivors that we went out and sampled. We have survivors from Fort Pierce, and we know from our data loggers that we had out there that the temperatures at that site were extremely high, 38, 40 C. This is like bathwater. So we don't know if the high temperatures killed the grass that died, but we know that the grass that survived lived through that hot temperature. And behind our property at our Florida Oceanographic site, the temperatures weren't as high, but the turbidity was very, very high. For months, there was very low light. And so I've written these or drawn these out for you so that you can think maybe the survivors at Fort Pierce might be a different strain than the survivors behind our property because they were exposed to different stressors. But that was just a hypothesis. So it very well could have been that all of these meadow strains survived, but just, fewer numbers. Or it could be that one strain survived in both sites. So we don't know. That's why we have to collect data and analyze it. So we ran a 2B rad experiment. We sampled from our source population in the meadow where people collected that grass and put out. And then we sampled our Fort Pierce survivors and our FOS survivors. And this is one plot of a lot of data. And I'm not going to show you all that. But what I want to draw your attention to here first is that Fort Pierce survivors are in red, the FOS survivors are in blue, and then the green is our meadow, our source population. And the first thing you'll notice is that there are meadow samples with no survivor representatives. So these are ones that are growing in our meadow, but when we sampled the survivors out in the wild, we don't find any that are related to these. This group over here are survivor samples that are nested within meadow representatives. And this shows that we can identify the most closely related stock plant because we know the location of each of the plants that we sampled. So for example, I can go to this green guy, M11-1A, and I know that this grass is the most closely related to this survivor that survived in Fort Pierce and this survivor that survived in FOS. And I know exactly where that plant is in my meadow, and I can go and pick it and grow it. And then there's this group over here, which is so fascinating. It seems like whenever I do research, there's always something that makes no sense at all, and it's really exciting when that happens. So these are survivors that were collected at both our Fort Pierce site and our FOS site that have no meadow representative. Now, it might be that we need to sample our meadow more, that maybe those guys are in our meadow, we just didn't sample them. But it might also be that these are wild recruits. So maybe these are samples that were, came back from the seed bank or something like that. So there's always more questions and more work to do. One thing I want to draw your attention to is that the survivor populations are closely related. So there's not two different lineages or two different groups, one with blue survivors that's most closely related to, one of our meadow plants, and then another one of our meadow plants being most closely related to the FOS survivors, right? So what that means is these survivors survived at both sites and are related to the same source plants, which is different than what I thought, right? I thought there was going to be, those green ones at Fort Pierce and the purple and the pink ones at FOS, and I was wrong. So that's really exciting too, to learn what's actually true. You come up with these hypotheses, and then you dig into the data and you realize it wasn't what you thought. This is a structure bar plot. It shows different subpopulations. And what our data suggests is that we have two populations. And they're colored here, blue and red. And the colors show the proportions of ancestry for both lineages. So some of the plants in our nursery are 100% one lineage, and some of them are 100% the other lineage. And then we have this mixture of others. And so one thing that's really fascinating to me is, so the red ones on this end are all survivors. So it looks like that the lineage that we've colored red in this bar plot, that lineage is the lineage of survivors. And then you have individuals with less and less and less of those genes. And so now we can dig in and figure out exactly what those genes are. So seagrass projects commonly fail because the IRL isn't healthy. And so we keep trying, and we change our methods, and we do new things, and we try again, and we try again, and we're going to keep trying until the IRL is healthy and we have beautiful seagrass meadows. But we need to learn all we can from these failures, and instead of getting really upset and sad and depressed, we need to stop and think, how is this failure an opportunity? What can I learn from this? And how can I apply it to the future to make our work successful? So here we sampled survivors from the field, from those mass mortality events, and we compared them back to their source population. So now we can intentionally propagate that source stock that was most closely related to the survivors. We know that our grass is made of these two lineages, and we need to study those to understand how those different lineages are interacting with stress. We can identify the genes that were selected for during that mass mortality event with this 2B-RAD data that we have. And we can build gene arrays to identify shoal grass with those resilience genes. So we can do that. We can go and survey the IRL and find natural populations that have these resilience genes once we know what they are. We can go to other nurseries and sample their grass and then say the ones in that tank have the resilience genes and the ones in that one don't. And we can do that in our own nursery as well. So this is work that I'll talk about next year. So after we propagate these meadow plants that are most closely related to the survivors, we can test them for resilience to different stressors. So we don't know what killed that grass. We suspect that the grass, we know that it's resilient to high temperature and high turbidity, low light levels. But we can do clear experiments to ask, like, how hot can they survive? Or, how long can they go without a lot of light? And once we know which ones are most resilient to those things, those stressors, then we can do intentional science-based restoration. And at this point, what we do is pull up the grass in our meadow and go plant it, and we do the best we can. But once we have this information, I can say the plants in that tank will do very well in this hot region, maybe near the shore where it's shallow and hot. And those plants in that tank will do well in this area that has high turbidity and low light levels. So that's my hope for the future. None of this would be possible without my super awesome team of researchers at FOS and my collaborators. I also want to say that frankly, everybody at FOS is awesome. We've had educators and people from the development team going out, doing the oyster monitoring or coming out to see what we're doing. They're always totally willing, to work with us. And the education team always highlights the work that we're doing and, calls us out and tells people about different research projects that we're doing. So we have a lot of support and love at FOS. Also, donations made to our team and made to FOS have been pivotal in this work. All of the water quality work was basically paid for by donations and the work that is done at FOS, people coming in and visiting and memberships and things like that. We have a wonderful supporters, and I'm very fortunate for that. And we also have lots of grant funding, and so I always want to point that out. And I have no idea how long I've blathered on for, but I'm happy to answer any questions if Zach doesn't pull me off with the hook.
I have a question with the dermo. Are you seeing that in other sea creatures, and have you seen that it's zoonotic?
Ooh, good question. Okay, so
Krista, would you mind repeating the question for our home audience?
That's what I'm going to do. So the question is about dermo and whether it's found in other creatures, and if it's a zoonotic. Did I say that properly? Zoonotic, yeah. So it's specific to, and Natalia, tell me if I'm wrong, it's specific to oysters, and humans can't get it. Is that correct? Okay. But if we eat oysters with dermo, can we get sick at all? No. Okay. I didn't think so.
And it's not in other animals?
No. Okay. But I do want to say there are more diseases that oysters get that we could have focused on, but for this work we focused on dermo. Did you have a question? No.
Other than overfishing oysters, what is the one problem that causes the
The problem that causes the most demise of oysters, well, that's a great question. So it depends on what time period you want to think about. So for the last, 100 years, we've been destroying oysters directly by pulling them out and over harvesting. And then now we recognize how important oysters and other bivalves are. And so we're doing less of that kind of direct damage. But the things that are really a problem now for oysters is water chemistry, water quality. And so we have, the discharges from Lake O hopefully are few and far between anymore, but that comes with a lot of other pesticides, nitrogen, phosphorus, things like that. We also have runoff from our yards that cause problems for water quality, and the pesticides that we use, the fertilizer that we use runs off into our lagoon. And so those things are current problems that we need to fix if we want healthy oyster populations.
That would include red tide?
So harmful algal blooms are a problem. And there's lots of different kinds of harmful algae that are are all around Florida. And they do cause problems for lots of wildlife. But those harmful algal blooms many times are linked to water chemistry. And so I would argue that we need to get the water chemistry clean. So the question is about what potential things the genes that we're studying in the seagrass might do. My suspicion is that it has to do with antioxidants. So antioxidants are proteins that help break down toxic chemicals. They help deal with oxidative stress, and they help cells deal with internal stress. And so I think that's what we're going to find are those potentially heat shock proteins and antioxidant genes.
Follow-up question to that. The survivors, do they have, I know you showcase different colors of grass to differentiate different subspecies or what have you. Is there a visible difference between the survivors? Are they like more green in this way, or is there anything visible about them?
The question is there anything visibly different about the survivor plants? And I think that's really a fascinating question because, man, wouldn't it be awesome if we could look at them and say, these have what they need to have, and they'll be resistant to, this slew of stressors. And the answer to that question is, I don't know yet. I suspect it's not going to be that easy, but we just got this data very recently, and so now we can go back out into our meadow and we can look at the ones and we can start asking those questions. I suspect if there's something that might tell us, I think it might have to do with whether or not, or the degree to which they senesce in the winter. I think what we're seeing in our meadow right now is a lot of seagrass senescence. That means it's dying back because it's cold. And so what we might find is that those grasses that are resilient to stress don't die back so much in the winter. So that might be a characteristic that we can look at. And wouldn't be very difficult. We just have to go out there and find, the tags for each of the grasses.
Krista, I'd like to grab a question off of our Zoom Q&A board here. What are the causes of the increase in dermo in artificial oyster reefs versus natural reefs?
So I don't have to repeat that one, right? I don't know. That's a good question. I think that it's likely has to do with stress, that the artificial reefs that we've put out are not natural reefs, and that the organisms that are settling there are stressed. Remember that the IRL is a lot saltier than the St. Lucie estuary, and so some of those oysters might be kind of at the threshold of their salinity tolerance, and so that could be stressing them out as well. And so that's why I think if we do these oyster reefs, we should put them in places where we know that's an ideal habitat for oysters, and rather than putting them places where they're kind of at the limits of their functional abilities.
I have a question about the bags that you're putting the oyster shells in. How did you pick the material, and are you ever getting like fish stuck in there or anything else?
So these bags went out before me. They were plastic, and so a couple years ago, people really weren't aware, or I think people were aware of problems with plastics, but there weren't very many good alternatives. I haven't seen fish getting stuck in them. And the idea is that, they're bags and then things settle on them and then build up the reef from there. And so for those first couple of months, maybe a year, little things can probably get inside, but over time, they're supposed to, build up the reef. Nowadays, people aren't using plastic anymore, and there's some really cool alternatives. There's alternative bags made from basalt, which is a rock. I don't know how that happens, but so it's super soft. It's like a net, and it's just a natural product that isn't believed to cause problems like plastic. I could do a whole talk on plastics and the problems that they cause for the marine environment. So we're all sort of on the same page now, where we're not doing plastic anymore. There's also, there's a company called Bees, and they make a bag, and it looks like plastic, but it's made from cast off potatoes from potato chip factories, and so it's starch based, and so it does degrade, over time, and they make different versions that last 2 years or 4 years, and you can choose, and so there's lots of alternatives now. Yeah. Yeah.
So you were talking about how you put seagrass and some had fences around them, some had not fences, and you talked about the survival of them. Do the manatees tend to eat more of the ones without the fences on, and how could that die off?
So the question is about the fences and whether those fences helped the grass survive manatee herbivory, so before I got to FOS, one of the projects that Lorraine and her team did was put out fences and tested whether or not that the grass grew better than having no fence. And they did actually find that the manatee ate the grass that was not fenced. Not surprising, right? And actually, she says they were waiting for the fences to be removed because a day after those fences came out, the manatee were all there. And so they're pretty smart. So one of the things that we do in this field when we're planting grass is we sow the grass onto burlap and then kind of stake that on the bottom. And the problem is that it takes a bit of time for the roots to grow and get established. So when the manatee are eating that grass, they can kind of pull it up really easy, even after a year. And so one of the things we're going to try is to plant sods. Now, this is like as if you're planting, you know, a plant you got from Lowe's or something, and, you know, it's that whole dirt pile, right? And plant that whole thing. And I think that's going to be, the roots are matted in there because it's been growing for a while, and it's also heavy. But that's also the downside, because instead of carrying 1,000 little mats of grass and planting 1,000 mats of grass, we're going to be carrying 6 sods, you know. So there's always pluses and minuses. So we're gonna try both of those this year and see what happens.
Unfortunately, time has flown by, and that's going to have to be our last question for tonight. Everybody, help me give Krista a big round of applause and a thank you. I know some of you still had unanswered questions. Sips in Science tomorrow night at Fraser Creek. So if Krista didn't have a chance to answer your question tonight, tomorrow will be a much more informal setting, short presentation, lots of back and forth conversation. We hope to see you there. And I hope to see you in 1 week for John Nelson's incredible storytelling all about the history of the Florida Everglades. Good night, everybody. Thanks again.